Viral diagnosis and treatment are in a lot of ways similar to bacterial diagnosis and treatment, but in a lot of ways very, very different. In terms of diagnostics, you might see some things on here that you recognize, including detection of proteins, detection of genomes, serology, growth, microscopy. But some of our differences are things like cytologic examination. We're not going to be looking at the viral particles themselves for the most part. We're going to be looking at the changes to host cells when those host cells are infected with viruses. The microscopy is also different. If you are going to try to directly look at viral particles, you'll have to use something like an electron microscope instead of a regular bright field or light microscope. Viral isolation and growth are also tricky, and we'll talk about why in just a minute. For specimen collection, you're really considering some of the same things as you would with collecting for a bacterial infection. You have to collect the samples hopefully early in the infection. The type of collection, of course, is definitely dependent on the area of the body that's infected. And it is important to get the sample to the lab before the virus degrades. Remember, we did talk about two different kinds of viruses, enveloped versus naked. Which type of virus do you think is going to degrade first? I mentioned those cytologic examinations, and what you're looking for if you are the clinical laboratory professional is distinctive changes to the cell that are caused by viral infection. And we call those changes cytopathological effects, or CPEs. These can include things like changes in morphology, lysis of the cell, formation of vacuoles, inclusion bodies, and syncytia. And syncytia means these kind of multinucleated cells. So it's a single cell that'll have multiple nuclei. Microscopy of viral samples is possible, but again, to actually visualize the virions or viral particles themselves, you have to use something like an electron microscope. Light microscopy simply doesn't have the appropriate resolution. There are two ways that scientists will use an electron microscope to visualize viruses. One is called immune electron microscopy. In this case, you're using an antibody to cause the viral particles to clump. And this is really useful when maybe the viral concentration within the sample isn't dense enough that you'd have to really be scanning and scanning and scanning to try to find viral particles. So you can use antibodies to cause them to clump so you can see them. The other type of electron microscopy is direct electron microscopy. And this is going to be a lot more useful when you have a sample that has really dense concentration of virus. That'll be something like rotavirus or even norovirus in stool samples. To culture viruses is really difficult because you have to be proficient in using any one of these systems. You can't just throw a virus on a plate and have it grow. So you have to be able to use animal models, different types of culture models. You're generally not going to use people, but people are a system of propagation for viruses. But yeah, animals, eggs, organ culture, and tissue culture are really the best and only ways that we know of to propagate viruses, because viruses have to replicate inside of the host cell. Some of the easiest types of cells to use are tumor or cancer cell lines, but the challenge with using those is, while it's a lot harder to kill them in a lab, right? You're not going to mess them up as much as you might mess up a primary culture line. Because they are cancer cell lines, sometimes they also look messed up under the microscope, and it can be really challenging to identify those CPEs because the cells already don't look normal. So for those of you who are going into clinical lab science, it's going to be really important for you to take classes in histology so that you know what normal tissues should look like under a microscope and what abnormal tissues will look like under a microscope. If you do end up deciding to do viral culture, and there are some viruses for which you could, a couple of the things that you're going to be looking for are shown in these images. So this panel on the top is a normal cell culture. I don't know what cell type it is, but it's a normal cell culture. And you see what we call a nice confluent monolayer. The cells are touching, but they're not growing up on each other. They're pretty normal looking. When you infect them with a virus, you start to see big areas of clearing. These are considered plaque formations where the infected cells have died. We also see these syncytia, these multinucleated cells. So these cells that are virally infected look really, really weird compared to the culture that's not. And that is what you, as the professional, are going to have to be able to identify. Some of the other cytopathic effects you can see here include cell death, those histologic changes, including the inclusion bodies, differences in how the chromatin looks, the syncytia, those giant multinucleated cells, and various cell surface changes. So why am I showing you this when I'm telling you you're often not going to be doing viral culture? So how are you going to see these things under the microscope? Well, you may be given a tissue biopsy from your patient. So you might be looking at an actual patient sample where you can see these cytopathologic effects. We can also quantify viruses. Again, this might not be something that you do in the clinical setting, but these are the terms that you would use if you do. So the titer is the dilution that retains the specific properties. And we have 3 different titers that we talk about. The tissue culture dose 50 is the titer that will kill or cause effects in half of your culture cells. The lethal dose 50 is the titer that will kill 50% of animals. And the majority of you will not be actually doing this type of work where you're infecting animals, but it is possible. There's also the infectious dose 50, the titer that leads to symptoms in 50% of animals. Those of you who might be doing viral culture in the lab may want to know something about plaque forming units. And that's basically, if you have a nice monolayer, plaque forming units are the number of host cells that have died. And that or become infected, mostly died, though you're looking for that clearance in the monolayer. And that represents a host cell that was infected by a single viral particle. However, when you're trying to do this, a negative result isn't necessarily conclusive because a lot of these viruses are difficult to work with. And also a patient can be shedding the virus without having symptoms. Or, for example, you could have, if you're doing animal work, animals that aren't experiencing symptoms but actually do have the infection. So viral quantification is really challenging, especially when compared to bacterial quantification. Most commonly, when we're trying to detect a viral infection, we are looking for proteins. And these could be either viral proteins, or we could be looking for the host to have produced antibodies, proteins, in response to the viral infection. Something like the COVID-19 rapid tests are looking for COVID-19 spike proteins found in your respiratory passages. But you could go to a clinic and they could even run an antibody test to see if you produce antibodies against COVID-19 proteins. So protein detection is really common for viral infection. Nucleic acid detection can also be used. I I guess, clinically acceptable COVID-19 tests are actually real-time quantitative PCR. So they're looking for the viral RNA, they convert it to cDNA and then do PCR to determine if you have an active viral infection. Because remember, looking for host antibodies doesn't mean the virus is currently infecting the body. It means that the body mounted an immune response at one time. Identifying viral proteins, like through the rapid nasal test, that is looking for the virus itself. But the problem with those rapid tests looking for the viral proteins is that they sometimes can't detect an early infection. So any of you who have had COVID and done those rapid tests, you most often do the rapid test because you've started to feel sick. So by then, you already have enough of the virus inside your body to cause you to experience symptoms. Whereas with the PCR-based test, the virus can be detected before it gets to a higher level to cause the symptoms. So it is more sensitive because it can detect a lower amount of the virus. Other viruses that are DNA-based can be detected by regular PCR. Those are going to be our two most common nucleic acid tests. When you are looking at host antibodies, if you find viral specific IgM, that indicates a recent infection because IgM is the first antibody produced in response to an infection. So if you have high levels of IgM, that means you're either still infected or perhaps it was recently cleared, but it's a very recent infection. You have what's called seroconversion when the antibody titer increases between the acute and convalescent phase. So the amount of antibody will increase because that's what your body does. It produces more and more antibodies during an infection. And so seroconversion is when you have a lot more antibody than you did at the very beginning of the infection. So some of those limitations with serology, again, it's not known exactly when the infection began. So if your COVID test turns up positive, your rapid test at home, how long have you actually had the infection? Could you have spread it to other people? So when it went through my house, the first person got it on Sunday, the next person turned positive on Tuesday, the next person was positive on Wednesday. I didn't turn positive until Friday, but I certainly, almost certainly was incubating the virus long before that. So it's tricky to determine when the infection began. You can have false positives, you can have some cross-reactivity, but you can also have false negatives if you don't have enough antibody yet, or if you don't have enough viral protein yet. It can show negative even though you are actually infected. So I was taking tests every day, Sunday, Monday, Tuesday, Wednesday, Thursday, negative, 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 negative. But at some point I did have the virus, even though the rapid test was showing that I was negative. Certain viruses can bind with antibody and serum. That can complicate the results. And of course, you have that cross-reactivity, things like parainfluenza and mumps. So it'd be hard to diagnose one of those infections using serology. Some are very, very specific. They have very, very specific antibodies like rhinoviruses. So there isn't like a standardized antibody test that you can do. In terms of viral treatment, treating viruses is a lot more difficult than treating bacterial infections because viruses take over host cell machinery. So in order to stop a virus, you often are stopping the host cell. However, we will talk about a lot of different viruses that have some very specific machinery, and those are easier to stop because you can target those specific viral proteins rather than host proteins. There are a number of different steps that some of these various viral treatments will stop. as we talk about the specific viruses and their specific treatments, we'll talk about how exactly those work. In order to control the spread of viral infection, I think we've all been living through this for the past 3+ years. Limit contact with sources of infection, wear gloves, wear a mask, quarantine, improve hygiene, sanitation, and disinfection. Try to make sure that everybody is immunized. And if everybody is educated, that really helps to go a long way to decrease high-risk behaviors. Things to think about. Envelope viruses are sensitive to environmental conditions. So, for example, COVID-19 is an enveloped virus. So, as simple as using, standard household cleansers can help to disrupt the envelope and prevent the spread of the virus. Non-envelope viruses are a little bit trickier, but they can be inactivated as well by things like ethanol, bleach, glutaraldehyde, formaldehyde, and autoclaving.